Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone. Don't forget, you are part of one of the most important scientific programs in the country, preparing future science leaders like yourself. In this phase, we won't just memorize biology. We will analyze, question, experiment, and think as scientists. Today, we begin a journey inside the smallest unit of life, the cell as it divides. Cell division is not a simple event, it's a precise, organized processes that keep us alive, and that we will see it and see it in Chapter 4, Cell Division. In a previous lecture, we had Introduction to Cell Division, and today we will speak about mitosis. Later on, we will see regulation of cell cycle and meiosis. Our lecture today focuses in mitosis, the division that produces two genetically identical daughter cells. The purpose of mitosis is to ensure each new cell receives a complete copy of DNA, and we will analyze each stage step by step to understand the full picture. Before we get deeper in mitosis, let's remind ourselves about cell cycle. Cell cycle has three main stages. The first one, interphase. The second one is mitosis. The third one is cytokinesis. Okay. Interphase here have three also steps. First one called G1. So first growth phase. In G1, it's begin after immediately cell division. So this is immediately after cell division. Also, the cell grows, organelles double, and it's perform its normal function. So it would be in normal, but with growth. Okay. Also, the cells spend the longest period. So G1, it's known as longest period on interphase. Okay. That in G1. The other stage is sDNA. And in sDNA or S phases, they called it DNA synthesis phase. Here what would happen? The cell's genetic material DNA is replicated. Okay? It's replicate and duplicate. Duplicate. Okay. This is in sDNA. Replicate and duplicate the DNA. The third stage is G2, second growth phase. This is proteins. What would happen? The proteins are synthesis here. And cells prepared for division. Okay, so that G1 is DNA, G2 is part of interphase. Now we will speak about mitosis. Now we have an idea. Before we go to mitosis, we are now in second phase of cell division. And in mitosis, we have four stages: prophase, metaphase, anaphase, and the last one, telophase. Okay, so let's speak about prophase. So first, the nucleus disappears because cell is preparing to reveal the chromosomes. Second, chromosomes condense so they can be controlled and moved. Okay. Also, spindle fibers appears like ropes that will pull the chromosomes here. When you see the spindle fibers here at the beginning that would rope and pull the chromosome, that immediately give us an indicate that we are in prophase, the first stage of mitosis. Move on to next step of mitosis is metaphase. So look at this picture, guys. Here cells reach maximum precisions chromosomes and it's align the cell equator. And if we want to examine chromosome, this is the best stage ever. We can see the chromosomes very clearly because they are in one line. Okay. And also in metaphase, they consider it as the shorter period in all mitosis. So in metaphase, chromosomes align along the equatorial plane of the cell. After we're done with the second stage, metaphase, now we will go to the third, which is anaphase. And in anaphase, sister chromatids separated and travel opposite the poles. Here, they are separated. One here and one here. And to be separated, the spindle fibers contract and be shortened to centromeres. Okay, and this happens quickly and preciously because errors can cause genetics abnormalities. So this is a critical moment of cell division. We reach to the last and final stage of mitosis, which is telophase. And in telophase, the cell begins to build two new nuclei for each set of chromosome. And the chromosome reach to the opposite poles, and the nucleus reappears, and finally chromosomes ankylate and return to less condensed state. Before the chromosome it will be very thick, and now it be to be ankylate and be less condensed state. This is the last stage on mitosis with telophase. This diagram summarizes the journey: condensing, aligning, separating, and forming a new nuclei. And look, guys, in top row, chromosomes condense and become visible. Chromosomes align at the middle of cell. Chromosomes separate toward opposite poles. Identical sets of chromosomes appear at both ends. In bottom row, nuclear envelope breaks down. Spindle fibers attack to chromosomes. Chromosomes are pulled by spindle fibers, and two nuclei begin forming around the chromosome sets. And to be clarify the difference between the top row and the bottom row. In top row, chromosomes changes. That's what illustrate. And in bottom row, the cell structure changes. That's the difference. After we done from interface and we done with mitosis, now we will begin in the third stage of cell division, cytokinesis. And after genetics material is divided, the cytoplasm separated to produce two complete cells. On animal, there's different on a plant. On animal, here, the membrane pinches, but in a plant, a cell plate forms. Here, cell plate, and here from centromere. Don't forget these main difference between animal cell and plant cell. And always cytokinesis The purpose of it is to divide the cell into two daughter cells, identical, because mitosis is to regenerate or to produce or to give us a cell identically. But in meiosis, no, there is a genetics changes. So in mitosis, the cell that will divide it would be identical to the parent cell. Let's examine ourselves in cytokinesis and mitosis with this question. The formation of a cell plate in plant cells occurs because centroles are absent, the nucleus is lateral, the vacuole is large, a cell wall is present. Think about it. What's the right option of these? Let's analyze the options. Let's start with A. A, centroles are absent. That one is wrong. Why? Because even though plant cells lack centroles, this is not the reason for forming a cell plate. So this is wrong. Let's move on with B. The nucleus is lateral. Also that one is what, guys? What do you think? False, is wrong. Because the position of the nucleus does not determine whether a cell plate forms. So this is also incorrect. Let's check C. The vacuole is large. This also wrong. Although plant cells often have a large vacuole, this does not explain the formation of a cell plate. So the C is incorrect. Finally, why a cell wall is present is the right answer of formation cell plate. Because cell plate form because plant cells have a rigid cell wall that prevent the cell membrane from pinching inward like animal cell. So the cell plate helps build a new cell wall between daughter cells. In summary, animal pinch inward, plants build a plate because of the cell wall. So the right answer would be D. Let's have another exercise. Which of the following is not true regarding mitosis in plant cells? Be careful, not true statement, false statement that we are looking for. So let's analyze the option. A, chromosome replicate during interphase. Why this is statement is true? Why this is true? And if it's true that we didn't need it to answer this question. Why this statement is true? Because chromosomes do replicate during interphase before mitosis begins. So statement A is true, so we don't need it. What about B? Spindle fibers originate from centroles during prophase. That one is not a true statement because plant cells do not have centroles. Spindles from because cells do not have centroles. Spindle fibers form without centroles. So that one is the right answer we need because this one is the false statement that we are looking for. But let's keep going and see and illustrate the other options. C. A cell plate forms during telophase. Why this statement is true? Because in plants, a cell plate forms in telophase to separate the two daughter cells. So that one is a true statement, and true statement we don't need it. Last one, a cell divides into two, each with the same number of chromosomes. Also that statement is true. Why? Because mitosis produces two identical daughter cells with the same chromosomes number. So this is a true statement and we don't need it. So the right answer B, because spindle fibers originate from centroles during prophase. And the reason plant cells do not have centroles, that's the answer. Let's have one more exercise. In which stage of mitosis can chromosome be seen and counted? Is it in prophase or metaphase or anaphase or in telophase? Prophase is wrong. That's wrong. Because in a prophase chromosomes begin to appear, but they are still not perfectly aligned, and counting them is very difficult. So we eliminate this option. Let's go to C. Okay, why C is not suitable? Because in anaphase, chromatids separate and move apart, making counting confusing. So we eliminate these options also. What about telophase? Telophase also is incorrect because in telophase chromosomes start to ankylate and disappear from few. So also we eliminate this option. So the right options go with what? Metaphase. This is the right answer. Okay? Because in metaphase, chromosomes align in the center of the cell, like we see here. Okay? Fully condensed and can be easily seen and counted. So the right answer would be B, metaphase. Okay, no problem. I will give you another exercise, but this is the final exercise that we take it today. How many rounds of mitosis must occur is in a signal cells to produce 128 cells? Is it 7 times or 14 times or 27 times or 32 times? Let me explain the key concepts of this question. In mitosis, each division doubles the number of cells, and this is the forms of sequences. 1 give us 2, 2, 4, 4, 8, 8, 16, 16, 32, 32, 64, and 64 give us 128. Okay, so let's remove the wrong answer. Let's do elimination. D, 32 is wrong. Why? Because 32 rounds would be produced far more than 128 cells. Okay, so D is removed. What about C? 27 round. This is also be removed because it still produces an extremely high numbers. What about 14? Also 14 is wrong. Why? Because if we do 14 rounds to produce more than, what do you think? More than 16,000. So that will be eliminated. The last one is 7 times. It's the right answer. Why? Because I told you before here. Look, 7 times. Because doubling 7 times starting from 1 give us 1, 2, 4, 8, 16, 32, 46, 128. So the correct answer would be 7 times. So it would be A. That's the correct answer. So today we learned how the cell divides and protects genetics material in the process. And we covered in this lecture mitosis, stage of mitosis, cell cycle, and cytoplasmic division, which we called it cytokinesis. Thank you for your attention. And remember, scientists begin with curiosity and continue with persistence. These organizations believe that investing in the mind is the strongest investment, like yourself. So you are the one who worth invest on him or on her. Thank you so much for listening. See you soon. Bye bye.